Thank you very much, Pat. And it will be really difficult to shift gears after the wonderful story that we heard from Rachel. But we are going to shift gears and we're going to talk about exon skipping. And well, I like to think of Pat and Elizabeth as my older sister, but and actually, so I'm chairing the session and I just thought of it. So next week, next Friday, I will be working on exon skipping for 22 years. So but when I started, I very naively thought, well, this is the approach, and then we'll develop something, and it will be approved, and it will be done. And I think what we realize now that it's not that simple. We know that the trick, so to say, works. We can do exon skipping, we can restore dystrophin, but there's room for improvement. And you will hear today 7 stories on exon skipping, on how different companies try to improve exon skipping in different ways. For the speakers, how it works, you each have 10 minutes. And what Pat didn't say, I bring chocolate, but I'm also the best timekeeper in the world. You have 10 minutes. I will stand up and walk there, and that means you have 1 minute left. So then you really have to wrap up. They installed a trapdoor just for me. So if you keep talking, that will happen. Then afterwards, you can sit on one of the chairs, and we will have a panel discussion at the end. And I think people will have questions, so therefore also make sure you don't run over time, because you're here to share your story, but of course you're also here to answer questions of the participants. So we're going to start with the first speaker, which, if I remember correctly, is Teji Singh from Sarepta, who will talk about the SRP-5051, which is a peptide-conjugated morpholino oligomer. And you are mic'd up, so the floor is yours, and the time will start ticking.
All right, lots of pressure with that time, but it's hard to follow that Awesomeness talk. But I do know I'm going to put Awesomeness reports into our clinical trials for assessments, and I'm going to use my Digitis Pendentis long-term to work this control. So thank you again for inviting me back, especially after the previous presentation this morning. I'm glad I actually got to come back in here. Same disclosure, I still work for Sarepta after that morning session. And like you saw from this morning, we're still actually working a lot in the DMD space. So in Duchenne's, we have 20 programs, 2 gene editing, 4 gene therapy, and today we're going to talk about our programs in RNA therapy, which we have 14 programs, both non-clinical and clinical. I'm going to focus on the 3 programs that we have in the clinic. They're PMO and PPMO. PMO is our phosphodiesterase morpholino oligomers, or those ASOs, and the other P for PPMO is our peptide conjugated PMOs. 3 of these are actually approved, 3 PMOs. They're approved for use for exon skipping of exon 51. That's eteplirsen. That was approved or accelerated approval in the US in 2016. We have exon 53 with goldemersen, and that was approved in 2019. And casimersen for exon 45 skipping, that was approved last year. Together, these 3 cover about 30% of the mutations and address 30% of the population. I'm going to take one little detour for the families that are here. If there's any questions about how to get access to these drugs and how to help you through the process, the contact at the bottom, the Sarepta Assist, which are patient advocacy group and our Sarepta colleagues can help you with. If you contact that number, that'll set you up with a case manager that can walk you through the complexities of the insurance, the complexities of the access and the healthcare systems. So that little plug out of the way, I'm going to just talk about our 2 late-stage programs in clinical programs for our PMO. And we have 2 studies that are ongoing. We have our Essence study and our Mission study. Our Essence study is an ex-US study, and this is for CASI and for GOLO. This is studying exon mutation amenable to 45 and 53 skips. There's 40 sites in this global study. The purpose of this is really to get the long-term data in terms of function after administration of CASI and GOLO. Mission is our study that's both in the US and ex-US for eteplirsen, and I'm going to go into more details on the next slide. This study is for 51 skip amenable lesions. It's also a global study with sites in Greece, Korea, Zealand, Spain, Taiwan, Turkey, and the US. So New Zealand, of course, being an awesome place to have a study site in. So mission, what is the purpose of mission? So our labeled dose for eteplirsen is 30 milligrams per kilogram. And what we wanted to do with this study is to see if we actually gave more, 100 milligrams or 200 milligrams, do you get more expression or more dystrophin or more percentage mRNA transcripts that are exon 51 skippable. So in this study, we did a quick pilot with 10 patients to prove that it was safe to dose at 100 or 200 milligrams. We had the non-clinical package that confirmed it was safe, and that part was successful. So we then kicked off the part 2 of the study, which is currently enrolling. In part 2, we want to have 144 subjects. They're blinded to treatment assignments, but they're going to be divided into 3 groups. The 30 milligram per dose, which is our labeled dose, 100 milligrams or 200. It's a 144-week study with the primary endpoint being NSAA, and patients will have 2 biopsies. Any individual patient will get 2 biopsies, the one at baseline and one depending we randomize them into which biopsy group they get, but we'll have a biopsy at week 24, 48, and 144. And this is basically to assess with different dosages, do you get more bang for your buck? Do you get more dystrophin? So that study is ongoing right now. Key things here, it's for ambulatory boys from an age of 4 to 13, the NSA being the primary endpoint, no placebo, and all participants will get one of these 3 dosages. Skipping a little bit ahead, no pun intended, that's our PMO2 studies. We're also doing a study in our PPMO. So we have one clinical trial, we have one clinical candidate that's in clinical trials, and that's our SRP 5051, or a eteplirsen. So basically it's a eteplirsen with the conjugate in front of it. We've done and completed a healthy volunteer study in here. We've done a dose finding study, which was our 102 study, and we've now started our part 1, part 2 study of phase 2 called Momentum. So in Momentum, what we're trying to do here is to look and see if this drug, 5051, actually leads to greater expression because it's the peptide conjugate. So basically here in part 1, Momentum part A, we actually demonstrated by proof of concept to show that giving a peptide conjugate does lead to enhanced percent microdystrophin mRNA transcripts. This is based off skin biopsies. That's that far graph on the left. And from that far graph, you can see in the 2 doses that we studied, the 20 milligrams and the 30 milligrams, that the higher dose led to more percent transcript production. The one on the right is how much is that related to dystrophin production. And again, you can see with both we get higher dystrophin production, but more with the 30 milligrams than the 20. Now keep in context that 3% dystrophin with the 20 and that 6.5% with the 30. This is more than what we have with the eteplirsen by itself. So that actually showed that the peptide conjugate actually did get more production, which was our theory. And the reason that we think that works is the peptide conjugate works actually by enabling more trafficking of the PMO into the target cell or the muscle cell, but not only more into the cell, but actually once it gets in the cell, it gets moved or trafficked to the right part of the cell so it can actually skip the lesion. So it's not just getting more in the cell, but once it gets in, it takes it or traffics it into the right portion of the cell. One of the things that we did, there was, again, just by chance, one of the patients was actually late getting a biopsy in this study for a variety of reasons, so ended up getting 5 dosages. That allowed us to check what had happened if you had a little bit more time on drug. Their expression was actually higher than the patients that got the 3 dosages just because week 12 was our biopsy endpoint. And we use that to kind of model what we'd expect long term. So we would expect the steady state for this drug roughly around 6 months. So by 6 months, we expect the expression to be higher, so 12, 15, or 16%, which we'll have to prove in part B of the study. Now, I'm going to stop for half a second and just go over safety. In this study, part A, plus also in our non-clinical program, we had demonstrated that with these, with the PPMOs, specifically this one, that due to the peptide, we did see some loss of magnesium. So you get something called hypomagnesemia. So this is usually mild, self-limited, goes away all on its own without intervention. But in part A, in the treated boys, we had one serious adverse event, which was a grade 3 adverse event of a low magnesium level. The patient had some symptoms with it, but resolved or responded with supplementation. That caused us to go back and look at the design of this study and put in some lessons learned into our part B, which is more intensive monitoring, all patients were going to get prophylactic magnesium. So you take that by mouth. It's usually well tolerated, but some people can get GI upset with it. And the idea here is that that actually prevents us from dropping it. We also noticed from our non-clinical work that we did around the same time that magnesium tended to be early, so with the first few dosages, and it tended to be when more pronounced at higher values than lower values of total dose. So we also looked here to make sure that as a run-in in our Part B, patients would get a low dose, a second dose, and then they would get randomized into the study, and I'll show you that design in a second. This was not associated, the low magnesiums itself was not associated with any kidney damage or markers of kidney injury. And if you could just file that away, after the next slide, I'm going to come back to the safety issue, because in Part B, we started the study with all these in place, but I'll tell you later that we actually had another safety event, and there's consequences of that. So Part B of the study, it's enrolling. It's about 1/2 completed. Basically here, this is both ambulatory and non-ambulatory boys between the ages of 7 and 21. We were looking for 60 participants. 30 of them were previously treated. They were in our phase 1 study, and 30 are going to be treatment naive, so they'll be brand new patients. At time of entry, they get a low dose, 350, another dose of 750, then they get randomized either into a high dose arm or a low dose arm. It's an open label study, so everybody gets treated and they know their assignments. We do a biopsy at baseline in week 28, only in the treatment naive, not in the previously treated ones. The endpoint of the study is actually expression. That's the primary endpoint, but we're following them out to 2 years to look at their functional state. So this is going well. Like I mentioned, it's half enrolled. But last week, we actually had to submit a report to the FDA about another SAE. This was a grade 3 SAE with a boy that was detected on our routine monitoring to have low magnesium. He has a few symptoms after he was detected, which is tingling, but likely due to another electrolyte. Was treated as an outpatient, just got magnesium supplementation. The magnesium resolved within a couple of days with the IV and then the oral supplementation. But the FDA came back to us and said, you need to submit more data about this case, and you need to reevaluate your procedures for testing. So we're on clinical hold in the US for this study until we submit them, which we plan to do next week. But we can still dose ex-US. The same package, same information was submitted to other regulatory bodies, and they haven't responded, but of any changes or additional information that we need. So in the US right now, we're on hold for this study, but again, we plan to submit our package next week and then follow it up. And seeing someone that's coming towards me, but I have a minute and I'm on track. That's it. That's the presentation. And if there's more to learn, please feel free to just ping this and catch us anywhere in the room today. Is that okay?
Thank you. Thank you very much,
And fairy fingers. So.
Teji, for this presentation. And also thank you very much for elaborating on the safety concern. I think many of us had seen the announcement online, so I think it's good that we have some more information now. The next speaker is Mike Panzara from Wave Therapeutics, and he will talk about Wave N531, which is an exon 53 skipper. The time starts soon.
Wow, thanks. I'm so nervous now just about that. Good afternoon, everybody. Thank you very much to Pat and the team for inviting us to speak. You know, I'm a neurologist, been doing drug development neurology for a little over 20 years now, but this meeting is really unique. It's really great to be back. It's very inspiring. This community is very inspiring. So again, thank you for inviting us to participate. I'm going to talk about N531. As was said, we all do exon skipping and we all have lawyers. That's what this is. So Wave is a genetic medicines company, meaning we target genetically defined diseases. So diseases where there is a genetically targetable problem. We have a unique approach to DMD, which I'm going to go through. We have an exon skipping approach, which I'll talk about in the details. We've initiated our N531 study in 2021. Many of you may remember that we actually had our exon 51 skipping drug fail in 2019. We've learned a lot about what happened with that failure. We've applied a lot of those learnings to this compound, which is why we're back and which is why we're forging ahead, and I'll talk a little bit more about that. And we have other programs in genetic diseases. So I don't have to spend much time on this. Just the concept here is restoring functional dystrophin through exon skipping. Now, our unique approach is that we have a chemistry and a toolkit that enables us to, as we're designing an oligonucleotide, which is a small strand of RNA or DNA, we're able to control different aspects of the oligonucleotide that others are unable to control. So most people, you're able to control the letters or the sequence. We're actually able to apply different types of backbone chemistries, meaning chemistries that actually only work in the context of the second aspect we're able to control, which is the orientation of the linkages, also called stereochemistry. And what we've been able to do is by using these tools, create what are called stereo pure oligonucleotides, so single strands with these modifications, that when we then bring it into our preclinical studies allow us to optimize for that single compound. And that allows us then to test and explore more preclinically, and then understand when we get to clinic the profile of the drug that we're bringing to clinic. And I'll go a little bit more through that. So N531 is an investigational therapy, exon 53 skipping. It incorporates the new chemistry that arose from some of the work trying to understand what happened with our suvodirsen program, our exon 51 skipping. This chemistry, when you apply it with stereochemistry, seems to get into the cells better. It distributes throughout the body better than this first generation stereopure molecules, which is what suvodirsen was. The molecule shows really interesting in vivo data, and we didn't have this for the suvodirsen molecule. We used a severe mouse model for DMD called a double knockout model, and we compared it to the first generation stereopure chemistry. And what we found is that this new chemistry improves survival, improves serum biomarkers, meaning the measurements of different effects of the drug in the tissues, and we really, unlike the other compounds, saw improved skeletal muscle and respiratory function. And that's what this slide is illustrating here. So what you're looking at here is on the left-hand side, you're seeing a graph. And on that graph, the lines represent animals that were treated with either a suvodirsen-like molecule, which is stereopure but doesn't have the PN. And the green line represents what an N531-like molecule. So it has stereopure and PN chemistry. So what you see here is the different lines are going down as mice die. So as the mice die, the lines go down. What we found with this new chemistry is that our suvodirsen-like molecule had a little bit of an effect, but the mice still were unable to survive. However, in this model, we found that the N531-like molecule actually improved survival to the end of the study at 2 different doses. And what was remarkable about this is, as shown in the box, the N531-like chemistry seems to make it so you can dose less frequently and at a lower dose. So that was really encouraging for us, and we did all this prior to making the decision to go back into clinic, because we wanted to make sure we had enough information about this new chemistry that it had at least the potential to improve over suvodirsen, because we didn't want to go into clinic unless we had a chance of having an improved profile versus suvodirsen. And then we looked in the tissues. What you're looking at here, this is the dystrophin amount that we measured in the different tissues. The other thing that was very interesting for us is you're seeing in the diaphragm and the heart, compared to the first generation chemistry, which is the blue bar, really improved uptake in the diaphragm and the heart, which is likely responsible for this improved survival in the mouse model. So this again made us reassured that this compound and the chemistry we're applying to it is different than suvodirsen, and made us want to take it into at least preclinical toxicology. And that's what we started to do here. We actually put it, first of all, then we took N531, which had that chemistry, and we basically tried it in cells, and you put cells in a dish, and we found exon skipping improvement there. We actually looked in a monkey, and we looked in the tissues of the monkey to see what kind of improvement we had in skipping. And then we actually looked at the half-life because in our first open label study. And what we're finding is that the drug lasts a lot longer. Well, suvodirsen, we found, disappeared very quickly. This N531 seems to have a half-life of greater than a week, which means half of it is still there in over a week, which really gives us reassurance that the effect of the drug may be longer, which gives us the potential to have a reduced dosing frequency every 2 weeks or longer. This is the study that we started in 2021. It's a small study outside the US where we just have a few patients that we're treating. Open label with the eventual goal of getting up to a dose that's well tolerated, and then there'll be a single biopsy, and we're going to look at where the drug is in those biopsies, because with suvodirsen we learned that the drug was all stuck around the muscle, not in the cells. Here we're going to look and see if the drug gets into the cells. If the drug is different from what we saw with suvodirsen, we'll expand the cohort and we'll continue with additional dosing with additional biopsy in a different patient population to see what do we do in terms of dystrophin. It's really a small start, but we felt this was the best way to proceed to see if we fix the problem that we had with our previous chemistry. So, went backwards. So, along the way, I just want to a couple of things. We incorporated a lot of the learnings from suvodirsen, but a lot of the learnings from suvodirsen were also from the community. We have no placebo in this study because in this particular scientific question we're asking, it wasn't needed. We actually have very limited biopsies in this, just for the purpose of being able to assess, is the drug different? And then we're planning to, as we go along, continue to communicate. After the suvodirsen study, we distributed all patients' data back to the families so that everybody was able to have their own child's data to understand how they did. And we, of course, commit to continue to take that approach along the way, and as we go forward, ask more questions. So basically, in conclusion, the N531 study underway, started in 2021 with data expected this year. We have preclinical studies and our initial clinical studies that the drug is well tolerated, suggesting a biweekly or less frequent dosing regimen. We're looking, obviously, benefit risk needs to be established. Current sites are in Canada and the UK, and then we will expand from there, potentially in the US in 2023, with clinical data this year. And just, again, the gratitude to the patients and their parents and people who contribute so much to enable us to do this, and trusting us after everything with our previous study to, again, partner with us as we try to move this next generation compound forward. Thank you. I do? It's okay? Yeah?
Perfectly timed. Thank you very much, Mike. We move on to the next speaker, which is Mark Stahl from Avidity Biosciences, and his compound is called AOC 1044, and I think the 44 goes for exon 44 skipping. And Avidity uses an antibody to improve delivery of the oligonucleotide to skeletal muscle. So the floor is yours for the next 10 minutes.
Thank you so much. Thank you to all of you for your attention, and to PPMD for the invitation to get to speak. My name is Mark Stahl. I'm a neurologist, and I'm from Avidity Biosciences. These are our forward-looking statements, which I will read to you in great detail. Avidity is a biotechnology company based in La Jolla, California. We consider ourselves a patient-focused rare disease company, with the ultimate goal to improve the lives of patients who are impacted by rare disease, patients and families, of course. And that includes Duchenne muscular dystrophy, through the development of new drugs, primarily those that deliver RNA in ways that have not previously been possible. We are currently working on several potential treatments for the DMD community using a technology that we developed in-house. Our technology has the potential to perform exon skipping, which of course we've been talking about today, in a different way by improving the delivery of the drug itself. And in doing so, we want to express our deep commitment to the DMD community in participating in events like this and hearing, really what is important to you and your families, and incorporating that into our work at every step along the way. And I'll tell you a little bit about where we expect to be able to bring our initial program, AOC 1044, into the clinic by the end of this year. So let me first begin by telling you a little bit about what our technology is and how it works. So our drugs are called antibody oligonucleotide conjugates, or AOCs, a little bit easier to say. AOCs are designed really to target specific tissue types. And in this case, of course, we'll be looking at targeting skeletal and cardiac muscle. And the goal, of course, in this case is to treat the underlying dystrophin deficiency that's present in those muscles. And so this is how we do it. We have two primary components of an AOC. The first is an antibody. As most of you know, especially over the last few years, we're all familiar with antibodies. But in drug development, antibodies are often used to target particular molecules. And those molecules can be on, for example, the surface of a tissue type that we think that we want to get a drug into. And so we use that property to bring our drug to exactly where it needs to be, to the muscle tissue. And so that's the sort of delivery vehicle, if you will. And then the oligonucleotide, or oligo, as you've all heard a few times already by now, is a short strand of DNA or RNA that really is the business end of the drug that does the exon skipping and changes the way that a protein is produced. And so by combining these two entities, we're able to make a drug that really goes where we need it to go and does so in an amount that has a significant effect. So we're working right now on multiple programs that are all exon skipping therapies, and our first project is AOC 1044, which is for patients whose mutations are amenable to skipping exon 44. And we expect to start our first clinical trial by the end of this year, with the other programs to follow. So as we've talked about a little bit so far, exon skipping is really established as an effective way of increasing dystrophin quantity. But as most of you know, there are some significant limitations in how effective it can be. And primarily, those relate to how quickly they're eliminated by the body, and then also how well they get into the muscle cells themselves. And so while once the oligonucleotide is able to get into the muscle, it works very well. We wanted to improve the actual delivery of the oligonucleotides into the muscles. And so this is where the antibody component comes in. This is a cartoon that really shows you a little bit about how we see this drug working. And so you can see we have a cartoon of an antibody that has multiple oligonucleotides attached to it. That antibody attaches to a protein at the surface of a muscle cell called the transferrin receptor 1. That receptor antibody pair is brought inside the cell in a process called endocytosis, where the antibody and the oligonucleotide are separated. And then the antibody is essentially degraded while the oligonucleotide is released to get into the cell and do the work that it does. So you can almost think of the antibody as like a bus that delivers passengers to the station where they need to go to work. And the passengers are, of course, the oligonucleotide that does the business of skipping exon 44. Of course, the end result of all of that is for the exon skipping to produce a shortened but still functional dystrophin protein, much like is seen in say, an in-frame Becker mutation. So I'm going to show a little bit of data. Unfortunately, this will mostly be from cell culture and from mouse models, but I think it'll give us a little bit of the idea of what we want to expect from our drug. So the first is that our oligonucleotide component is able to achieve significant amounts of exon skipping in cells that were taken from patients who have mutations that are amenable to exon 44 skipping. And so we can see that our oligo works very well on its own, by itself. But of course, the real question is, how well do we do compared to with the drug as a whole component with the antibody attached to the oligonucleotide. And so in an experiment where we used a single dose at the same equal doses of unconjugated oligonucleotide or naked oligonucleotide versus the same oligonucleotide at the same dose attached to our antibody. And so you can see we were able to obtain a little bit of skipping, detectable amount when we just gave the oligonucleotide by itself. But when we use the antibody to affect the delivery, at the same amount of oligonucleotide, we were able to see an increase in skipping of about 50 times, which we feel will lead to significant amounts of dystrophin production. And so, of course, the next question is, how much dystrophin did we actually get? And so, again, these are single doses given to a mouse, where we're able to see, again, in comparison to control, we're able to see significant amounts of exon skipping, and then again, we're able to see dystrophin protein restoration on that right side panel in multiple muscle tissues. So with good distribution to different muscles, we're able to see large amounts of dystrophin being produced after only a single dose of our AOC. This next slide, I think, is really important to see and really helps to demonstrate the power of the AOC with a very nice, even, homogeneous distribution of the dystrophin production. And we feel this is important because it keeps the function of the muscle consistent throughout the muscle fiber, with dystrophin being expressed, very evenly throughout. And we feel this is really, in many ways, because of the active delivery of the drug into the muscle. With this scientific background in place, I wanted to really just kind of finish up by saying how much we are committed to working with the people here and the rest of the DMD community to bring forth, treatments that are meaningful for you. And we are incredibly interested in hearing, what you have to say in terms of giving us your input. So please reach out to us here and through our email address that is, there you go, at the bottom of the slide here, with any questions or things that come to mind with our treatment. So thank you again very much. Appreciate it.
Thank you, Mark, and yeah, please take a seat. So the next speaker is not here in person, and I learned yesterday that the reason he's not here is because he's celebrating his wife's 50th birthday. So I think one of the few reasons where we say, okay, you're allowed to skip a PPMD meeting for that. We have a pre-recorded video from him, so it's Ash Dugar from Dyne, presenting on Dyne 251. And well, hopefully the video lasts 10 minutes, and I understood that there's also some Q&A at the end. So I'll give a maximum of 11 minutes, and then I'll ask you to stop the video if it lasts longer. So please start the video.
Hello, I'm Ash Dugar, the Global Head of Medical Affairs at Dyne Therapeutics. First, I want to thank the organizers of the PPMD meeting for a wonderful conference and for inviting me to speak today. It's always a privilege to present to you. Today, I'll talk about Dyne's Force scientific platform, the newest data from our Duchenne program, and our clinical development plans. These are our forward-looking statements for your reference, and you can find them on our website. At Dyne, it all starts with our mission. We are laser-focused on delivering life-transforming therapies to patients with serious muscle diseases, with Duchenne being a priority. Our mission is core to who we are and what we do every day. Our focus today is on sharing some non-clinical data from our Duchenne program and update you on our clinical trial plans. We've built a robust portfolio focused on rare muscle diseases. DM1 and FSHD represent opportunities to develop first-in-class therapies. And in Duchenne, we're building a global franchise starting with our Exon 51 program. And we will follow this program with exons 53 and 45. This slide describes our FORCE scientific platform. And there are three components to this platform. There's an antibody, a linker, and then the oligonucleotide. And in the case of Duchenne, it's a phosphorodiamidate morpholino oligomer, or a PMO. So our FORCE platform consists of a specific delivery mechanism called a Fab, or an antigen binding fragment, or an antibody fragment, that we've engineered to bind to a receptor called transferrin receptor 1, or TFR1. This receptor is ubiquitous. It's found across the surface of muscle cells, including skeletal muscle, including diaphragm, and cardiac and smooth muscles. The Y-shaped structure on the left is what a full monoclonal antibody looks like. Now, our FORCE platform uses the top piece, or the Fab, which binds very tightly to the TFR1 receptor on muscle cell walls. We believe Fabs have several advantages over monoclonal antibodies, including its smaller size, which helps with better distribution to muscle tissue. Now, the Fab acts as a kind of key that unlocks the TFR1 receptor and allows our therapeutic antisense oligonucleotide, or ASO, to enter skeletal, cardiac, and smooth muscle to target the genetic basis of the conditions at the level of the nucleus. We link our Fab to the potential therapy with a linker, shown in this burnt orange string at the bottom. The linker molecule connects the Fab to our ASO, which gets in the nucleus. As I mentioned, in the case of Duchenne, we use a type of ASO called a PMO, targeted for exon 51 skip amenable mutations. In the case of other muscle conditions, we can use other antisense oligonucleotides. So this platform is modular and highly versatile. Historically, the challenge has been the inability to get enough of the PMO into the muscle cells to result in significant dystrophin restoration. Our FORCE platform is designed to solve the delivery issue and deliver the exon skipping therapy into muscle cells using this receptor-mediated approach by attaching the PMO to our Fab targeting the TFR1 receptor on muscles, and thus enables targeted delivery of the PMO to muscle. On this slide, we show our FORCE platform in the MDX model, a standard animal model in Duchenne. Now, these MDX mice have a mutation in exon 23. On this top half of the slide, you can see FORCE resulted in robust and durable dystrophin production across key muscles, like the quadriceps, the diaphragm, and the heart, with a single dose of 30 milligrams per kilogram, and we evaluated the mice 4 weeks later. We were able to achieve high levels of dystrophin expression, approximately 50% in the quadriceps, 90% in the diaphragm, and nearly 80% in the heart. We've even seen out 8 to 12 weeks durability of effect. And we are extremely excited to see these levels of dystrophin production, especially in the diaphragm and heart, where other modalities have had limitations. On the bottom panel of this slide, we show dystrophin localization in the sarcolemma after a single dose of 30 milligrams per kilogram. This localization at the muscle membrane is important to see to provide confidence in potentially seeing functional benefit. And we saw profound results, including about 70% dystrophin-positive fibers in the quadriceps and approximately 80% in the diaphragm and heart at 4 weeks. Next, we evaluated Dyne 251, our clinical candidate in non-human primates, or NHPs, or monkeys. These studies are a key step in generating preclinical data before going into clinical trials. In the study shown on the left-hand part of the slide, we evaluated exon skipping in monkeys after 5 weekly doses of 30 milligrams per kilogram of Dyne 251. We measured exon skipping in the quadriceps, heart, and diaphragm. Please note that in NHPs, we cannot look at dystrophin restoration because these are healthy animals. It is not a dystrophic model. The key message is that with our FORCE platform, time on therapy matters. When you look at this data in monkeys at month 2, we observed robust exon 51 skipping across muscles. And again, I would point to the heart and diaphragm. After 5 weekly 30 milligram per kilogram doses, Dyne 251 demonstrated 52% exon skipping in the diaphragm, 43% skipping in the heart, and 18% in the quadriceps. We also evaluated Dyne 251 in a 5-week good laboratory practices, or GLP, toxicology study in non-human primates. We saw that Dyne 251 had a favorable safety profile with no dose-limiting toxicity observed after 5 weekly doses up to a maximally feasible dose. Importantly, we pushed the dose many folds higher than the 30 milligrams, and we're very pleased with the safety profile as we advance to the clinic. Now, as we prepare to start our Dyne 251 clinical development program, we have gained expert feedback. We've engaged with the patient community and key opinion leaders, or KOLs, from various disciplines, including pediatric and adult neurology, cardiology, pulmonology, pain medicine, and physical therapy. Importantly, we've gathered this feedback across the globe. And KOLs both in the US and outside of the US, including Europe, with experience running early and late stage trials, have provided advice on our overall clinical development program, including the clinical trial design for our first study, looking at the patient population, biomarker, and functional endpoints, as well as key safety considerations, and have imparted learnings from natural history studies. It's clear that KOLs believe dystrophin continues to be a key endpoint for this disease. We've also received advice directly from patients and caregivers, as well as global advocacy leaders, such as those from PPMD. They provided insights based on their living with and being the voice of the Duchenne community, as well as experience with past and current trials. This input has been critical in ensuring a patient-centric design and has included key considerations for choosing a clinical trial, feedback on biopsies and functional outcomes, identification of the most burdensome or difficult parts of the trial, and how to ensure patients and families are supported throughout the process. Based on this extensive feedback, we've developed the proposed clinical study for Dyne 251 in patients amenable to exon 51 skipping, and I'll describe some of the key elements here. Our clinical trial for Dyne 251 will be a global, multiple ascending dose study, or MAD study. It will be placebo-controlled with a long-term extension. We plan to enroll patients with Duchenne who have mutations amenable to exon 51 skipping therapy. Our goal is to enroll approximately 30 to 40 patients aged 4 to 16 years of age who are either ambulant or non-ambulant. And we plan to evaluate the following outcomes in this MAD study, such as safety and tolerability, dystrophin, muscle function that is relevant to both ambulatory and non-ambulatory participants. And our goal is to offer monthly or less frequent dosing in the clinic. We've also received questions from the community. The most common question is this: what's happened with our program in terms of the IND application? So please note that we submitted our IND to the US FDA in December of 2021. As many of you may have seen, we communicated to the community that the FDA placed us on clinical hold. We also sent a letter to the community about this event. We understand that the term clinical hold can be very concerning to the Duchenne community, given the history of clinical holds for other potential therapies in development for Duchenne. It's important to note that Dyne 251 is not yet in the clinic. Hence, this clinical hold is not related to an incident in the clinic or in a Duchenne patient. This clinical hold asks for additional clinical and non-clinical information about our program. So we're executing on our plan to respond to the FDA's request in the second quarter of this year. And if satisfactory to the FDA, we plan to begin dosing patients by middle of 2022, as we have previously communicated. And more details about our study will be available closer to when we begin dosing patients. So I'd just like to end by thanking you for your time and attention. Please know that we're committed to understanding the community of people living with muscle diseases, and Duchenne in particular. And we want to ensure that your perspectives and experiences are included in our drug development programs. This is central to our work each and every day. And thank you again to PPMD for the opportunity to present to you. I do wish I was there in person, and thank you so much for the time today.
Okay, well, I don't think he can hear us because I understood he's in Italy. But let's give him a round of applause anyway. So the people in the room from Dyne can tell him that the talk was appreciated, and that I think it was good that he had some Q&A in there at the end. So we're moving on. The next talk is from Nerissa Kreher from Entrada, and she will talk on 60144, which is an exon 44 skipper. They also have a peptide, but it works in a slightly different way from the one from Sarepta, the one from Dyne, the one from Avidity, and I'm sure that Nerissa is going to inform us about that. So the time starts soon.
Thank you. First, thank you to PPMD and thank you to the Duchenne patient community for welcoming Entrada. I'm Nerissa Kreher. I'm the Chief Medical Officer at Entrada Therapeutics. As many have shown you, we're a public company, so here is our disclaimer statement as well. So let me first tell you a little bit about Entrada because we are relatively new. We are a company that is driven to help patients that have devastating diseases utilizing a technology we call endosomal escape vehicles, and I'll explain to you what those are in just a few minutes. We're a Boston, Massachusetts-based company. We have about 120 employees now. We are committed to developing treatments for people living with devastating diseases. We're very focused on our company culture, and that translates to also the culture of how we interact with patient groups like PPMD. So this commitment goes through many different things, and it first starts with listening to the patient community. So I'll tell you one example of how listening to the patient community has driven our decision-making at Entrada. As we got into the oligonucleotide space and the Duchenne space, we were thinking about what exon we should first target. And in conversations with groups like PPMD, we have made the decision to first focus on exon 44, so developing a therapy for patients that are 44 skipping amenable. And I'll be talking to you today about that. It's called ENTR 60144. So just one example of how listening has helped driven our decision making, and you can fully anticipate that that will continue. So why are some of us up here working with peptides that are conjugated to oligonucleotides? Well, it's related to a problem we have in drug development related to getting drugs into the cell. So about 75% of all disease-causing targets are inside of cells. And we haven't been very good at getting the drugs to those targets. At Entrada, we think that we have a solution to doing that. And it's a twofold solution. So the first is getting it into the cell. And the second is avoiding a clearance mechanism that often can shuttle drug back out of the cell very rapidly. Those two things end up that you don't get enough drug to see the kinds of effects that you really want to see. So the current problem in the Duchenne space, of course, we are seeing drugs that increase dystrophin slightly, but we think that we can do better utilizing our EEV technology. So let me tell you about that technology. The EEV is a small cyclic peptide. So you'll hear from some of my colleagues today that are working with linear peptides. Ours is actually circular or cyclic. And so that's what you see, the little yellow cartoonish blurb. And that's linked to the oligonucleotide, which is the exon skipping piece, again, which you've heard some of my colleagues talk about. So that cyclic peptide allows us to get a lot of drug into the cell. It enters the cell easily. And then importantly, it's not cleared out of something called the early endosome. So the drug buds out of that early endosome and then is available in the cell to reach the nucleus and cause exon skipping. In the next few slides, I'm going to share some of our data with you. I'm going to start with some of our mouse model data. We actually use a mouse model called the D2-MDX model. So this is a very rigorous model of Duchenne muscular dystrophy. These mice have very bad muscle disease. On the top part of the slide, you see exon skipping. And so what you're looking at is our drug, EEV, combined with the oligonucleotide, and then compared to vehicle, think of that as like placebo or water, and then the oligonucleotide conjugated to nothing. We sometimes refer to that as naked oligos. The teal bar is our EEV conjugated molecule. And you can see in both the heart, diaphragm, and an example of skeletal muscle, we're getting very high exon skipping. In the skeletal muscle, up to about 100%, and in the heart, about 60%. As you all well know, the heart and the diaphragm are critical muscles in Duchenne muscular dystrophy, and seeing exon skipping and dystrophin production in those muscles is really, really important. On the bottom part of the slide, you're seeing the actual dystrophin production. So the dystrophin is the green fluorescent color. On the far left-hand side is what we call a wild-type mouse, so a mouse without disease. In the middle is that vehicle treated, so you see very little dystrophin production. The next panel is the naked oligo or naked PMO. See a little bit of dystrophin production, you can see some green. But as you can see in the one treated with oligo conjugated to EEV, you're seeing a lot of green. Very, very similar to what you're seeing in the wild-type animal. Lots of dystrophin production and very homogeneously distributed. So this is exciting to us. We think that this will mean that it can translate into dystrophin production in the heart, in the diaphragm, and in other skeletal muscles. Now, importantly, that's great, we have dystrophin, but we would like to understand if that dystrophin leads to improved function in the muscle. And we have some evidence in that same D2-MDX model that that's the case. So creatine kinase, you're probably familiar with. It's something that's released when you have muscle damage. And so what we would anticipate in a diseased model, there would be high CK or creatine kinase because there's a lot of muscle damage. So if you look, it's kind of hard to see our teal bar. That's because the CK is low, indicating to us that there is reduced muscle damage. The muscles aren't releasing that creatine kinase. Similarly, we're able to measure how the mice actually, how their muscles function with wire hang time and grip strength. So we're really testing how strong these mice are. And again, as you can see here, in both of the teal bars, we're getting good muscle strength, higher than the naked PMO, and very, very similar to the wild type or non-diseased animals. So we're seeing exon skipping translating into dystrophin, translating into functional improvement in this rigorous mouse model. So now we'll switch gears to the non-human primate or a monkey model. Now in the non-human primate, I think as was mentioned before, we can't measure dystrophin because these are not animals that have disease, but we can measure exon skipping. So in this experiment, we've given the monkeys one single intravenous dose of ENTR 601-44. On the left-hand side, we're measuring exon skipping at 7 days after that treatment. And you can see multiple skeletal muscles, quadriceps, biceps, diaphragm, and heart. And we see very significant exon skipping in the non-human primate model as well. Something that was really exciting to our team when we got these data in on the right-hand side was the actual duration of effect of exon skipping. So again, this is just with one single dose. We do serial biopsies in the animals, and as you can see, out to 12 weeks, we're still able to measure exon skipping. What we think that means is that in the clinic, we will be able to dose at intervals of every 6 weeks or possibly even longer. So really important as we think about the current therapies being weekly intravenous dosing and some of the ones in development being monthly, we're thinking we have the opportunity to dose at a longer interval. So as we think about our current clinical development plan, we're not yet in the clinic. We plan to file regulatory documents in Q4 of this year that would allow us to move into the clinic late this year, early next year. We plan on doing our first study in healthy volunteers, and then we will move into studying patients with Duchenne muscular dystrophy. We look forward to keeping the community updated on our progress. And send thank you and hello from Boston. This is some of our team members, lots of people who contributed to the science I presented today. So please come and find us if you have questions. We love to talk to patients and families and look forward to continuing a relationship with the community. Thank you.
Thank you very much. Next speaker is Mellion from PepGen, another peptide conjugated oligonucleotide, this one targeting exon 51. So the floor is yours.
Thank you. I was running up here because I want to make sure I can use all of my time. My name is Mellion. I am the SVP and Head of Clinical Development at PepGen. Similar to everybody else, this is our disclaimer. We probably copied it from each other because all the lawyers know each other, and these are my disclosures. So for those of you that are not familiar, PepGen is a clinical stage biotechnology company. We're located in Cambridge, Massachusetts. And similarly to what my colleagues have described here, what we're looking at is how can we get these oligonucleotides to the target most efficiently with maximum exposure. And so we took the approach with our enhanced delivery oligonucleotides. And what we mean by an enhanced delivery oligonucleotides is that these are well-characterized therapeutic PMO oligonucleotides that are conjugated to a proprietary delivery-enhancing peptide. So some of you may be asking yourself, what the heck is a peptide? A peptide essentially is a string of amino acids. And what our peptide looks like is it contains 3 domains. 2 of the domains in dark blue are called arginine-rich domains, and they have a very big positive charge. And this is separated out by non-natural amino acids. And these 2 domains are then separated by a hydrophobic core. And by taking this peptide and putting it together with the therapeutic oligonucleotide, so this is the genetic medicine that we've been talking about that targets the root cause of disease, we have our enhanced delivery oligonucleotides, which we believe will result in efficient cellular uptake of oligos, including in the cardiac as well as in the skeletal tissue. And so our pipeline really, as you can see here, our lead program is an EDO51, but also we are very committed to looking at other exons in the DMD space. So we're building out our portfolio for DMD by looking at 53, 44, and 45. And we also have a lead program in myotonic dystrophy. So today I'm going to take you through our MDX mouse data, the non-human primate data, as well as share with you our clinical development plan, and also see how we can further engage with you, the community, which is pivotal to the success of any clinical program. So in our MDX mouse data, what we did was we dosed the MDX mouse with 1 dose of, it's called EDO51 is how we refer to it. And you can see here that on the left, we are showing exon skipping. On that horizontal axis, what we are showing is the different muscles that we looked at 7 days after giving this single dose to the MDX mouse. And we looked at striated muscle or skeletal muscle, that being quadriceps, biceps, and the diaphragm, and importantly, the heart. And you can see on that vertical axis that we're looking at the percent of exon skipping. And NT means no treatment, and then the 30 and the 60 is the dose that we gave to the mice. And you can see here that with increasing doses, for after just one dose, there's an increase in exon skipping across all tissues. Additionally, when you take a look at that graph on the is that on the right? Sorry, is the dystrophin. So again, on the x-axis, we're looking at those same tissues at those same doses. And now on the y-axis, what we're looking at is we're looking at dystrophin. And how we report out dystrophin is the percent of normal or of wild type. And you can see that in all of these tissues, there is a dose dependent, meaning that there is an increase in the amount of dystrophin that is being produced across the tissues, up to 99% in that muscle, and notably 25% in the heart, which is critical, as well as 80% in the diaphragm, which, as you all well know, is critical and is part of the cause of mortality for our Duchenne patients. But, you know, as mentioned previously, in these MDX mouse, we have to look at a surrogate. We're looking really at skipping of exon 23. So it's important for us to look at our clinical candidate in our cousin, the non-human primate. So we're able to look at exon skipping with EDO51 in the non-human primate. And after giving a single dose of EDO51 in the non-human primate, we looked at the exposures or the concentration in each of these tissues. So specifically skeletal muscle, and we took representative tissues, and you can see here on the horizontal axis, again, what we're showing are the tissues that we looked at, as well as we're looking at the different dosages which are shown in those different colors. And on the y-axis is the concentration that we are seeing in each of these tissues. And again, you can see here that there is a dose-dependent increase in the concentration across all of these tissues. But importantly, we're also getting into those hard-to-reach tissues, that being the cardiac tissues, the diaphragm, smooth muscle, as well as we are seeing some exposure in the central nervous system. Additionally, we looked at the results of exon skipping in the non-human primates after giving them 3 doses of EDO51. So these 3 doses were given every other week, and we looked in these different tissues after that 3rd dose. And so you can see here in the blue bars, which are representative of EDO51, and here on the x-axis or the horizontal axis is the dose. So the dose was given at 0, 10, and 30 to the non-human primates. And on that vertical axis, again, is the exon skipping, the percent exon skipping. And we looked in the quadriceps and the biceps. And again, we see this dose-dependent increase in exon skipping. But not only did we look at exon skipping with EDO51, we also compared it to the most advanced PPMO that is currently in development, and that is shown in the purple bar. And what you can see is that at each of these doses, EDO51 showed more exon skipping than the most advanced PPMO that's currently in development, as well as you can see that at 10 mg per kg of EDO51, there was approximately as potent as the 30 mg of the R6 Gly, which is the most advanced PPMO in development currently. So at 1/3 the dose, we're getting the same amount of exon skipping. Additionally, we looked again at the cardiac or at the heart muscle, specifically the left ventricle, and we also looked at the diaphragm. And again, on the horizontal axis is the dose, and on the vertical axis is the percent of exon skipping. And you can see here that we were able to see that there was exon skipping to about 23% in the heart, and about 75.9% in the diaphragm. And again, you can see that there is a difference between the R6 Gly as well as EDO51. We also, what's not shown here is that we did look at exon skipping in the heart at 20 mg per kg. This was after a single dose. And looking across the whole heart, we saw about 20% exon skipping in the whole heart in the non-human primate. We also took samples after we gave each of these doses every 2 weeks. So again, on the horizontal axis, you have the days. So, and these are the days in which we gave those 3 doses biweekly. And we took essentially biopsies of quadriceps and biceps at day 8, 22, and 36. And we looked at exon skipping. And looking at exon skipping levels, what we saw was that exon skipping actually accumulated with repeat dose administration for EDO51, and that this accumulation over time was greater than the R6 gly. And they look like they're not going to cross. They're essentially parallel. So we anticipate that we will continue to accumulate exon skipping with repeated dosing. And this data gave us confidence to move into our phase 1 study. We currently have an ongoing phase 1 study in healthy volunteers in Canada. And some of you may be asking yourselves, why are you doing a phase 1 study in healthy volunteers? And we feel that this data is very important because it'll give us an idea of the safety and tolerability in healthy volunteers. So this basically helps us understand that tolerability profile. But importantly, it also helps us to understand the penetration or how much EDO51 we're getting into muscle, and also give us an idea of the efficacy. So obviously we're looking at healthy volunteers, so we're looking at exon skipping, but still this can give us an idea of efficacy, so that when we go into the phase 2 study in DMD patients, which is planned for the first half of next year, we will be able to start at potentially more clinically efficacious doses and skip over those homeopathic doses. So our anticipated development plan is, as you can see, we are already in humans. We are planning on sharing this phase 1 data at the end of this year. And we plan to start our DMD trial in 2023, and we'll analyze that data in 2024, and plan to meet with you and clinicians and others to determine our path forward. So it's important that we continue to engage with you, and we're going to be looking to you as we continue on this development journey to be sure that we are developing protocols that are meaningful to you and the DMD community, and that we can move together to understand what the best therapies are for Duchenne muscular dystrophy. I thank you for letting us present our development program to you today, as well as to the healthy volunteers and to the study staff that's executing our phase 1 study, and to PPMD. Thank you.
Thank you. So we've made it to the last presenter in the session, which is David Jacoby from BioMarin, and he will talk about BioMarin 351. So the floor is yours, and you have 5 minutes. Sorry, you have 10 minutes. No, you have 10 minutes. And then there will be time for questions, and I know that there's also online questions coming in. So feel free to think of questions, because we'll get there in 10 minutes.
Great. So Dave Jacoby, I'm also a neurologist. I'm one of the clinical developers at BioMarin. It's a pleasure. It's always a pleasure, but it's great to be back. I'm very thankful for the invitation to speak about BMN 351 and give you an idea of what BioMarin's plans are for development in Duchenne in the future. Maybe a quick opportunity to speak about BioMarin. We are a more established biotech company. We've been around for 25 years. We're 3,000 employees worldwide. We have 7 approved products and continue to grow, expanding both the types of genetic challenges we'd like to approach, but also the modalities with which we do it. The development philosophy with us isn't any different than my colleagues and panelists up here on the stage. We're a genetic medicines company, and I think you'll see that the problems of Duchenne map beautifully onto the philosophy of clinical development that we have at BioMarin. We're very interested in targeting genetic diseases, ones in which we understand how the genetic lesion translate into loss of function or gain of function that we think we can target with direct genetic correction. So the point here is we're not interested in nibbling around the edges here. We're trying to find targeted therapies that really correct the genetic deficit. We think if you do that, there will be a large effect size, something that is easily measurable, and that's going to transform basically the lives of patients going forward. So that's the ethic, the philosophy of development at BioMarin as it is with the colleagues up here on the panel. like others, our development program actually has evolved from a development setback, and that's drisapersen. So we actually worked with the University of Leiden and a small company called Prosensa to do the pivotal development of drisapersen in 2014. And when we submitted that data, basically it was judged not to have the risk-benefit profile to favor patients. And so that was the point at which we sort of thought about what our philosophy for doing science in Duchenne was going to be. We've seen some really elegant science today about one approach, and one approach is the reason these skippers are failing is because we're not getting enough of them into the muscle. So if we can target more into the muscle, we're going to have a better chance of skipping more message and getting more protein and having a better effect. And I think also it's worth saying that all of us up here really believe that full-length or near full-length dystrophin given back to muscle at reasonable doses is more likely to have a functional benefit than a micro or a mini dystrophin. So all of us are sort of bound here to think that the exon skipping is really a great approach to solving at least those proportion of mutations that are amenable to those skips. But the approach that we took wasn't to sort of see if we could get more drisapersen into muscle, was to see whether there was a better molecule than drisapersen. And so our scientific approach was to actually tile through the areas that might lead to more potent oligonucleotides that allowed skipping of exon 51. And so delivery is great, but we believe that there's a steeper dose response. Basically, if you can identify a molecule that has a very strong potency for its target. And RNA processing and RNA metabolism is a very tricky thing. And so we believe more potency ultimately is going to result in more skipping and more protein. So that's the sort of fundamental difference in our approach compared to my colleagues up here on the panel. So we have identified an oligonucleotide that has significantly more potency at skipping exon 51, and we're in the process of working that up into a clinical candidate and going into the clinic probably next year. Yeah, so my slide build here. So let me talk to you a little bit about BMN 351. So you've seen a lot of numbers and a lot of data in experimental models, and what I'm going to show you is the actual clinical candidate. It's not a tool compound. It is the exon 51 skipper, and this is a mouse model in which there's the human gene for which the exon 51 skipper can actually make a near full-length dystrophin. So it's called the DEL52 mouse. This is an experiment in which we dosed with BMN 351 in this mouse model for 13 weeks. And then we assayed for dystrophin protein 1 month and 3 months after we stopped dosing. So the data that you see here is basically dose for 13 weeks, look for dystrophin 1 month after and 3 months after. So one of the things you can actually see here is the molecule is really quite stable in cells, and once you make dystrophin, it's a very stable protein. But you see with 2 doses, a low dose and a high dose, that we're getting sort of 20 to 40% and 30 to 70% low dose and high dose in the peripheral muscle, and that is stable between 1 and 3 months. There is no decrement amount of dystrophin that's there after you finish dosing. And if you look in the heart, this is a different scale. So, but typically with naked oligos, we see maybe a 5-fold difference between peripheral muscle and heart. Here, we're just seeing about a 2-fold difference. But you see significant amounts of dystrophin protein produced in the heart, both at the low dose and the high dose, 1 month and 3 months after finishing dosing with 351. And then lastly, I agree with our colleagues up here that dystrophin levels are great, but what's the translatability of that, and how do we think about what levels are responsible for what function going forward? So the luxury of the DEL52 is that there is a motor phenotype that's testable. We use this motor rater score, which is actually far more complicated than I can explain. It's a sort of this high frame videography that looks at the mouse in terms of its stride length, the pace of its gait, its posture, its coordination and fine motor abilities, and it sort of aggregates that into a single aggregate score. And basically a higher score is worse. So you see these mice basically as a function of time accrue more disability. The scores go up. And the low dose here essentially stabilizes the motor rater score. But really reassuringly, the high dose here actually reduces the motor rater score to that of a wild type, a normal mouse. So you're actually seeing here a degree of reversibility of a phenotype in a fairly severely affected mouse. So we're excited about not just the dystrophin levels, but also the relationship of the dystrophin levels to a functional readout in a severe and we think relevant transgenic mouse model. Okay, well, is that the last slide, truly? Yeah, I guess it is. So let me just give a quick update. This is our clinical candidate. We are currently in GLP IND-enabling studies. We plan to file the IND later this year. So we hope to be in the clinic shortly after that, maybe early next year. We, like everybody else here, travel in a pack. So there's plenty of BioMarin folks here. So if there are specific questions that we can answer, but let me just also just say thanks for inviting us. And as a sort of a member of BioMarin, we're really grateful to be back and to be continuing development in Duchenne. Thanks.
Thank you very much for staying on time. And while people walk to the microphones, I can start with a question for everyone, because I know that this is probably one of the questions that many people have. What about other exons? And I know some of you told us already, but maybe you can say the additional exons, so what about them preclinically, clinically, on the list? So we'll just start with Teji and then you can make your rounds.
Sure. So 51 already in the clinic, 45, we have 53. We also have plans to do 30 that's in preclinical, and we also have 2 more that are in preclinical, depending on how the 31s go. So we have 5 altogether, preclinical, 1 in the clinic.
So, you know, as we enter back into the area, we're starting with 53, but, you know, our approach has been that as we learn about the chemistry, we're finding there are certain chemical structures and signatures that tend to, with stereochemistry in the PN, tend to carry across different sequences and different targeting. So the idea would be if we can show that this improved approach works for 53, we would then move on to multiple at the same time, because then you can apply these chemical principles across the oligos. So that's where we're starting, and then we would anticipate, again, going into the more rare pools of types of deletions.
Okay.
So we are starting at Avidity with Exon 44, as I described, and with ongoing work in an Exon 45 skipping. And as the information and data from those programs starts to build, we're going to continue to go forward with choosing the rest of the exons that we think that our technology will be most suitable for. So where we're at.
Nerissa.
Yeah, and I can use a lot of Mark's answers. So we are starting with 44. That's our lead program. We are looking at a candidate for exon 45 skipping next. And then we will be, looking at what makes sense from the standpoint of the next exons that we would choose. Again, informed with input from the community.
Thank you.
I think similarly with all my colleagues up here, the goal is really to try to treat as many people living with DMD as we possibly can. So I'm sure that we're all looking at any of the possible exons for skipping. So as I had said, currently with our lead is in 51. We do have activity as well as in 44, 45, and 53. And certainly we'll be working with you, the community, to identify other needs as well. But the goal is really to not leave anybody out, is really to treat everybody.
David.
Yeah, no, we're a little different in that we don't have a delivery technology, which is hook up the next oligo to it. So we're trying to understand a little bit about the science of what makes the site that we've discovered in exon 51 so potent, so that we can think about whether that applies to other exons and other mutations as well. So there's a little bit of just going back to the table and trying to understand the science before we move forward. And we're trying to tile, and we're trying to actually do pull-down experiments where we understand what part of the splicing machinery we're affecting with the new site. But the other thing to maybe mention is that in research here, in early research, we're looking at other ways besides oligonucleotides to modulate gene expression with the goal of trying to get full-length or near full-length dystrophin into muscle, more global treatments.
Okay, thank you. And there's a question there.
More and more of the boys are going into gene therapy studies. What will they be eligible for these various technologies, such as these various projects? I'm specifically referencing 44, but that's selfish. But I mean, just generally, if the boys had gene therapy, will he be eligible for all these developments that you're describing?
I see Mark nodding, so then you can take it.
I think that's a great question, and thank you for asking it. And I think that, I think we can imagine a day where perhaps having a gene therapy with a degree of microdystrophin production as a base on top of having an exon skipping with a larger, more nearly full-length dystrophin could be a great solution for getting the best efficacy someday. But I think in the near term, while we're doing trials to understand an investigational therapy, unfortunately they really do need to be tested individually. And so at the level of clinical trials, generally speaking, we would say that if you've had a gene therapy already, that you would not be able to be in an exon skipping trial.
The logic for that is what?
I'm sorry?
The logic for it, the medical logic for that.
Right. So the ability to understand exactly what the effect is of an individual treatment, it really has to be done, sequentially, essentially, or in isolation to understand what is really happening with that drug. But with that said, one thing that we didn't really talk about in discussing exon skipping type treatments, as in contrast to gene therapies, is that eventually, although we all want our treatments to last a long time, to have a long dosing interval, they do eventually become eliminated from the body. And so it may be the case that a person who is treated with an exon skipping drug at some point in the future could potentially do a different trial, at some point. Whereas that may or may not be true with a gene therapy.
Anyone has any? Yeah.
I would just add to what Mark was saying, that it's not only showing the efficacy of the drug, but also the safety, right? So if you've got two drugs on board, it's hard to tease apart where the safety signal could come from. So we have to be cautious about that piece as well.
Thank you for that very nice bridge to one of the questions that I'd written here, because Teji mentioned there was some safety concerns with his peptide, and I know the transferrin antibodies, that's a completely different thing. But Entrada is using a peptide, and PepGen is also using a peptide. So can you maybe share, do you anticipate similar safety concerns, or do you anticipate different concerns or less because your peptide is different? So maybe starting with Nerissa and then.
So as I mentioned, our peptide is different from others because it's cyclic. And because of that, the chemistry is very different. And so one of the things about the linear peptides we've heard is they have a lot of something called arginine on them. And because of the cyclic nature of ours, we can use fewer arginines. We think that will lead to a safer compound. We still, of course, have to test that and continue to test that in animals, as well as ultimately we'll move into clinical trials. Thus far, we have not in our animal studies seen issues related to the magnesium. But we will continue to follow that very closely.
Thank you. Michelle?
So I think from our standpoint, we're learning about the peptide technology, and so certainly when we have learnings with peptide technology, we all take it into consideration and go back and look at our own data, which is very important. So there's nothing more important than safety. And so for ours, we do see some similar safety signals across as this is a class of drug related to the magnesium at our very high doses that we believe are not within our clinically efficacious dose or that are beyond our clinically efficacious dose. So we anticipate that this was likely not going to be an issue for us, but certainly these are issues and data that we have to take into account as we're continuing to develop these therapies, and we'll need to be cognizant of them as we move forward with our development plans. And so the nice thing is that I know that it looks like we're in competition up here, but in actuality, we all learn from each other, and it's very important for us to share information so that we can all learn from each other and be sure that most importantly, we're putting the safety first and that we're able to put in plans into place where we can monitor that.
I see everyone nod. I understand we have questions online.
So there's a handful of questions, and the first one has to do with a question regarding an exon skip that's amenable to 61. So I don't know if that means a deletion of 61 or a skip of 61 to repair a mutation. Also, are multiple exon skips possible? And then the last one is regarding those who are on current exon skip therapies who are being treated. Will they be allowed to be in, what's the washout time? What would they be allowed to be in trial?
Okay. Anyone wants to take that? I'm getting my my exon sheet.
Sure. Well, I can start with the
For
last.
those who want to know, because so exon 61 is in frame, so we're going to assume it's not a deletion. We're going to assume that it's a small mutation within exon 60 with the information that we have. So anyone wants to say anything about exon 60?
That's you.
Yeah, you took the last one. As I said, we'll have to, as we learn more about what chemistry is associated with what effect and what pattern, we would basically try to expand. So it's hard to say at this point in time from our standpoint. And then in terms of in general, and I'm sure this is the answer for everybody, is people on current exon skippers, it's all about the pharmacology and the clearance, and meaning how much is left in the body. And unlike, as Mark was saying, the gene therapy, which can be longer lasting potentially and stay on board, the current exon skippers tend to wash out fairly quickly. So we've always allowed people on existing therapy, if they want to switch, they can and enter the study. We've never had a problem with that.
So if I hear you all correctly, then choosing beyond, say, the top 4 that now everyone is focusing on, because the groups are large, and you need large groups to show efficacy, the next exons will be chosen not just because of how large the group is, but also how easy it is to skip that exon, how well the oligo would work and be structured.
I think it's a very good point, Annemieke, that you're raising here, is that the reason we always go for the common exons is because it allows us to answer the scientific questions a little quicker. When you have a larger population, it's a little bit easier to tell, right? Quicker, potentially. And then you could expand. And then if you go into, from there, as you put groups of exons, that people with similar exons together into more of platform or basket type approaches, that's the next generation here, and that's the way it would have to be. And it's actually there are ways of doing that, and that's sort of where.
But if you're all going after the same one, at the largest group, you're all competing after that same group. So I guess the question from the community is, why is it just a financial decision? The community can take honesty. Is it simply a business decision at the end of the day, or is there a scientific reason behind it?
If it was a business decision, we wouldn't necessarily go after the one that everybody else is going after, right? Because the business decisions are how much are you able to eventually in the end sell. And that's, if we, Exon, let's use Exon 53. It's there, with multiple drugs targeting Exon 53 in the US market, it's not exactly the best business decision. Nonetheless, you are still able to, because there is, there's still an unmet need in terms of finding higher levels of effect, it's still possible to do studies with these. And outside the United States, it's definitely feasible to do, still target these exons. But I can tell you that, every company is different, but if it was going to be a business decision, the unmet need lies in the, all the other exons that aren't being
But as a follow-up question, can I then get, so as an academic, I would like for the next exon, these really small groups, that you do align, and that if there's 10 patients worldwide, that it's not 4 companies going for exon 35, but that you do decide, like, my chemistry is probably best for this one, our chemistry is probably best for, so I see people
Well,
not.
as Dave was saying, I mean, they're looking for very specific aspects of the sequence that may be very specific for some of these exons that we don't even know yet, but the research being done to try and address that is important. And there's also the question about multi-exon skipping.
Yeah.
That is that is a challenge that we've looked at, and as we develop new improved chemistry and new improved sequences, it becomes more and more possible.
Yeah. Filippo has been standing there forever.
Thank you. Exon skipping creates a new sequence that's not present in normal dystrophin. I wonder if you've looked to see if you're getting an immune response to that new sequence.
So I can say from our preclinical data, looking in the animal models, we have not seen that there has been an immune response. So there are ways to look for that in the animal models, and that has not been present. But certainly, to your point, that is something that we look at and include and still think about in the design of our clinical trials, and certainly evaluate that through our clinical trials. And so we'll be continuing to gather data, and I'm sure that all of you are also doing that as part of your clinical trials as an endpoint to make sure that we are not causing an immune response.
And maybe something I can add, because you're all nodding, and it's really good that you're monitoring it, but with the microdystrophin, there are new epitopes that are never seen by certain patients. So there really is a risk that it's a new protein. Well, actually, most Duchenne patients do low-level exon skipping already by themselves. So it's not new. They have seen it. They just do it at a very low level that is sub-functional. And so I think with that in mind, it would be, we wouldn't expect it. But that doesn't mean that it can't happen, and it doesn't mean that we shouldn't look for it. So it's really good that you look for it. But I think if you compare it to microdystrophin, the risk, in my personal view, would be lower. Do we have more questions online?
I have another. So and this might be for you, Anamika, actually. If I was a 51, my head probably would be popping off. These are mice. I don't know if I'd be able to make sense of linker technology, StereoPure. It's coming down to having a conversation with your clinician, rolling the dice and hoping for the best. I mean, I think it's probably hard for the 51s as well right now.
Yeah. So this is a question that has come up in Europe as well, because the clinicians are overwhelmed. They say, well, we get approached by multiple companies to do an exon 51 clinical trial, and well, what should we do? And also, we want to offer our families a choice, but what choice should we give them? How should we prioritize, and how should we do it? And the World Duchenne Organization is actually trying to get people in the room together to have a discussion like, well, what are the pros, what are the cons? And if you have different sides that, well, one side can get this and the other side can get that, the travel distance is not that far away, then people can still approach whatever they want to choose. And then I think it really is a personal choice, because there's like the eteplirsen high dose. Well, we know about eteplirsen. It's been used for a long time. People have received it for years and years and years. So the high dose is, well, it's probably not that risky. Well, the SRP 5051, we now know that there might be some safety concerns, but it does seem to work better. it seems to induce more dystrophin. And then I think it's a really personal choice what families want. Do they want the safe but maybe less efficient? Do they want, well, the safety concern but probably more efficient? And the challenge is that all of this, there's a lot of speculation and there's uncertainty. If we knew it would work, we wouldn't have to do the trials. But I think it's really important that the clinicians sit together and agree, like, we need to all have the same story, because if patients, parents, families hear, well, we go to that clinical site and they say, you should do this because this is best, and the other side, no, that is crap, you need to, that's not going to help anyone. And I think, if these drugs were not promising, they wouldn't be in the trial. So all of them have that promise, and well, that's why the companies are preparing for the trial. But which one is best for you, that's going to be a personal decision. And that will differ for each family, what is the best one. So I think that would be my recommendation. And there's work in progress, as you know probably, to try and get some advice on, well, this is the advantages, this is the disadvantages. And we would prefer one golden one that does everything well and has no side effects, but sadly that's not the case. We have more questions online.
So there's a clarification. The person asking about exon 61, it is a deletion. That is
61,
the mutation.
not 60. Okay.
Yeah, and anticipating another question that it regularly comes up is, there is a common variant in the Becker community of a very large deletion, and they're still preserved strong muscle strength. There's a lot of preserved ability. And is that a limitation with the technology that we don't approach that, or are there other reasons why that is not something that is explored? Because it would possibly help correct a lot of problems for those who have deletion mutations in that region.
So I'm going to start. I'm going to use the chair's prerogative and my 22 years of exon skipping experience. So first of all, 61 deletion is going to be really difficult, because you would need skipping of both exon 62 and 63. So you would need double skipping to restore the reading frame. And then you're approaching the area where the protein has a crucial domain. So I think it would be challenging from two aspects. So I think that's the sad reality. Then for the larger deletions, so actually this is something that I naively proposed, I think, in 2004. Why don't we do multi-exon skipping? Why don't we generate these huge deletions? Because many exons or many deletions will fall into it, and we're done in one go. But this is really difficult, first of all, because you need to skip a lot of exons. And then, so you need a ton of oligos. You need a ton of different oligos. You need to mix them all together. And then only if you manage to get all these different oligos to one cell will you get your effect. Most often, if you have a combination of 20, maybe 1 or 2 or 3 or 4 or 5 or anything, any combination will make it. And sadly, the structure of the dystrophin gene is such that if you have this combination, the majority of effects will be out of frame, so not functional, and the minority will be productive. So I think that's one of the reasons why, well, at least we stopped developing it. But there's a separate reason, and that is if you have a deletion and you make a mix of oligos, then that means that part of that mix is not going to be effective. So if you have a deletion of exon 50, for instance, you say, let's just make a 45 to 55 deletion, because we know it results in mild Becker. There's a lot of mutations there. Let's just do this. If you then have this mix of 45, 46, etc., until 55, for the patient with the deletion of exon 50, that means in that mix there's one oligo that won't work because he doesn't have exon 50. And the regulators don't like it, that you give something to a patient where you know in advance it's not going to work, because you know it's probably also going to have side effects. So you give something where you know it's not effective, it's not going to be beneficial, but it does have side effects. And I think that is a challenge of the multi-exon skipping that I didn't realize when I started proposing it back in the olden days. But I think for now, this is really a big challenge. So we need to find other ways to overcome it. But as such, it's not going to work.
I think what you're raising is a really important point, and it goes to one of the questions that was asked earlier. You know, on a piece of paper, the diagram of exon skipping looks very easy. You know, and maybe not that easy, but you cut out one exon, skip it, and things come back together and everything's great. But actually, as was being alluded to, skipping of different exons is easier or harder. And so we have to understand that, and that's part of the choice we're making as well. So I think it's just worth making that point that different exons may skip more easily than others.
And I think we're already seeing that for the 51 versus 53. 53 is an easier exon to skip than 51. I think we've run over time. That's what you get when you get me talking about exon skipping. But we have a last burning. So your question is the last question, and then we'll end the session.
Oh, okay, thanks. Will you be looking at rare exons that are in frame?
Rare in-frame exons. In Dutch we would say crickets.
I think it goes back to the answer that many of us have pointed to. Again, there are a lot of steps we're taking in making the decisions about each exon that we might choose to skip. It has to do with what I was just mentioning earlier, harder, easier. And so I think that's really a company by company decision and exon by exon.
No. Okay, so thank you all very much for staying on time and you for the questions.